Hi friends, so now I'm back with my next video, and in this video I'm going to talk about two different methods, FRET and FRAP, which are used to check the cellular dynamics like the protein-protein interaction, protein transport, the diffusion of different molecules from one compartment to the another using fluorescence microscopy. So don't forget to like, share and subscribe this channel. So what is fluorescence? So if you know this diagram, this is the Jablonski diagram. So according to this, suppose a molecule is in this state, the ground state, which is of the lowest energy possible at the steady state in equilibrium. So when a light of higher magnitude, electromagnetic light, comes, so it gains photons and then it excites to the excited state. So you can see from the ground state to the excited state, at this state it has tons of energy. But then, because of it has tons of energy, it is not in the steady state. It is in a non-equilibrium state. So the molecule tried to come to its equilibrium state, the steady state, by releasing this extra energy. So it releases this extra energy, could be here, but then it starts to release more. And the energy and the photon that are released at this stage is what is called fluorescence. So for different molecules, the photon release are different, and based on that, they have different lights. Like it can be for green fluorescent protein, which we see green light. For red fluorescent protein, it is red light because the energy that is released is different. And for each fluorescent protein, the energy that is required is also different. So now we know how this fluorescence works, and we can see the excitation, which energy which is required to move from the excited state, from the ground state, and the emission energy which is released to come back to the excited state. So what is FRET? How fluorescence can be used by this technique? So FRET is also called the Foster resonance energy transfer or the fluorescence energy transfer. So as the name suggests, the energy transfer means the transfer of energy from one state to another. So what happens is a transfer of energy from an excited molecule which we call donor. So here you can see the donor molecule. It has a specific excitation which is required to move from ground state to the excited state, and then it has an emission energy, the energy that is released when it is coming back to the ground state. And then there is another molecule which is called the acceptor molecule, which also have a certain excitation and also have a different emission. So energy can only be transferred between two molecules, like from donor to the acceptor, only if the emission of one is equal to the excitation of the another one. So here if you see that a molecule is excited at 435 and the emission is like 469 nanometers, and also for the acceptor molecule the excitation is 469 nanometers. So if these two molecules had the same emission and excitation, this emission which is released, like the energy that is released, it can be used here to excite this molecule. And in short, you can see it's like a is equal to B and B is equal to C. Here we can see that A is excitation wavelength and B is the emission wavelength. And if we go to the acceptor, B is still the emission wavelength and B is the excitation wavelength and C is the emission. So which means A is equal to B is equal to C is A and C combined. So here you can see the donor molecule, we see the excitation and we see the emission from the acceptor molecule. So you are thinking that okay, in the normal scenario there can be so many molecules which can have the same emission and excitation, which means we will always see the energy transfer. But what will that tell you? So this technique in which energy is transferred, it depends on certain conditions. So first condition I have already told you that the excitation of one and the emission of the other one should be the same to transfer the energy. And the other one, it only occurs when the distance between the acceptor molecule and the donor molecule is very small, which is like few nanometers. And if you can look here, there are two molecules, the donor molecule and the acceptor molecule. But the distance between, so So they are the excitation, as you can see, this is the emission and this is the excitation of the acceptor. It's not the same. So as I have already told you, so if the emission of the donor one and the excitation of the acceptor is not same, there is no FRET, there is no energy transfer. The energy transfer will only occur if the emission of one and the excitation of the another one overlaps. And if we see this overlap, it will cause the energy transfer. And the other thing that I have explained you now is the distance. So suppose these are two molecules each which they have the same excitation and like the emission of the donor and the excitation of the acceptor. But they are too apart from each other that the energy transfer cannot occur. So if the distance between them is more than 10 nanometer, there is no energy transfer. And if only if the excitation and emission are same, the distance is less than 10 nanometer, there will be energy transfer. And also the another thing that is important to consider is the orientation. If the orientation of two molecules is not correct, then there will not be FRET. Only if the orientation is correct, you will see the energy transfer. So now the question is, why do I care if the energy transfer is happening or not? So, yes, we care because this technique can be used to study different things. Suppose protein interaction. Suppose I label one protein with one molecule, suppose GFP, and the another one with YFP, which is another fluorescent protein. And I want to see if protein A is interacting with protein B or not. And suppose I give the excitation energy only for green one. And if these two proteins are interacting, I know that the emission is same, and only if they are interacting, the distance between them will be less, and only then they will transfer the energy. And because I first in the first scenario, I excited only with the green light, and if they are interacting with each other, the green fluorescent protein and the white fluorescent protein, the emission that I will see is yellow only if they are interacting. Because if they are not interacting, the emission that I will see is only for the green protein, not for the yellow protein. And only if they are interacting, the energy will be transferred from green to yellow, and I will see the yellow fluorescent. The same with the ligand receptor. I can label one ligand with one protein and receptor with another protein, the fluorescent protein, and only if they are interacting, the energy will be transferred, and I will see the excitation for one and the emission for another. And molecular co-localization, where you can see same like if the two molecules are going to the same compartment and to the same place or not by checking the energy transfer. So I hope this is clear, and if not, you can ask me in the comment section your doubts. And now I will talk about FRAP, which is called fluorescence recovery after photobleaching. So as the name suggests, what you do, you photobleach the samples. What I mean by photobleaching? Photobleaching is, as I explained you, the fluorescence where you excite your sample with the laser light. But if you fluoresce your sample, like if you excite your sample with too much energy, sometimes the sample get destroyed and it get bleached. So instead of seeing any color, you will see the black area there. And as the name suggests, suppose I want to study something, uh, diffusion or the movement of one protein from the another. Suppose I want to study the movement of one protein from the nucleus to the cytoplasm. But in the cytoplasm, the protein is already present, and I will not get better idea from where like how much protein is moving, and if it is the protein that I am seeing is already present, or is it moving upon some stress condition. So what we can do is we photo bleach that area, and once we photo bleach that area will become black, and all the molecules that are present there will become distorted, or there will be no protein. And then I will leave the sample for some time, and then I will see if the protein is moving from the nucleus to the cytoplasm or not. So how we can do it? We can label the protein that we want to see in the nucleus with fluorescent markers, And then we see the movement of this fluorescent microprotein from the nucleus to the cytoplasm. And in this way, we can study the movement protein in the living cell. So here is an example. You can see this is a lipid bilayer membrane. So here you can see I selected this area where I want to see the movement if the if you know in the lipid bilayer there are different lipids which are present. So I can see if the lipid bilayer is like diffusing, it's mobilized or not, or how can different lipids are arranged and if they can move between each other, because upon stress condition and upon the transport of some protein and passing through some endothelial, this lipid bilayer has to be flexible. So what we did is we photo bleached this area. So you can see this black dot is all photo bleached, and then you leave this for some time, and then you start to see the movement of this and then you start to see the movement of this lipid bilayer from one place to the another, and in the end, obviously the black area has been fully diffused. So you can see here I bleach my samples in the graph, and then you see then the protein starts to recover. Obviously, the intensity is less as compared to in the beginning. As you can see, in the beginning, the sample was at this threshold, and now it's at this threshold. Because I photobleach some sample and it get mixed, but it is not completely removed. So another example you can see here, this is the sample with all the actin, and this is one cell line. And you can see the normal sample, and then I selected this area which is called, which I want to bleach and which I want to remove. So this is post bleaching, and then we wait for some time where we want to see if the protein from the other compartment are moving to this area or not. So you can clearly see the fluorescence recovery. So this technique we can use to see the diffusion of different protein molecules and different lipid molecules. So suppose you want to study the signal trafficking, so molecular trafficking, you want to see how it works. If the protein is moving from the endoplasmic reticulum to the Golgi, from the nucleus to the cytoplasm, from ER to the mitochondria, what you can do is you can photobleach that protein or photobleach that area in the compartment and then see if the proteins are moving from the another compartment or not. So in this way, you can do molecular trafficking, intracellular transport, and to study the diffusion of proteins, lipid molecule, like I have explained now. Thank you. Please like, share, and subscribe my channel.